Information and Communication Technology presents Educational Satellite Television Programs.
Hello, teacher. Hello, students. Welcome to today's lesson. In the last lesson, we learned about methods used to control harmful microorganisms. We learned about sterilization, disinfection, and antiseptics. In this lesson, we will learn about different heat techniques used in sterilization. We will also learn about aseptic technique, inoculation, and incubation. Let us begin the lesson with an activity. Students, as a review, can you please explain the process of sterilization? Write your answer on a piece of notebook paper.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. If you answered that sterilization is a process that removes all microorganisms from the material or surface of an object, then you are correct. An object can be sterilized by applying heat, chemicals, high pressure, and filtration. Heat is one of the most common and most efficient ways to control the spread of microorganisms. Provided that the surface or the object is resistant to heat, this method can sterilize or reduce the number of microorganisms that are present. Students, can you think of something you or your parents do at home that uses heat to remove microorganisms? Take a moment to write down your answer.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. Were you able to think of an example? A common use of heat to remove microorganisms is in cooking. Certain foods, especially meat and eggs, must be cooked over heat to kill off the microorganisms that could potentially hurt you if you eat them. There are a few ways that heat can be applied. The first we will talk about is the simplest method, which is boiling. By placing an object in boiling water and keeping it there for some time, the harmful microorganisms will be killed off. Most objects need only about 10 minutes in boiling water, but some bacteria and viruses may take several hours of boiling before they are killed off. Autoclaving is another way to use heat to get rid of microorganisms. This method can only be used on materials that are not damaged by heat, such as equipment or supplies which are used in a laboratory. An autoclave is very similar to a pressure cooker, which you might use in your home. It boils water at a high temperature to create a high-pressure steam. The higher the temperature, the more force the steam has to kill off all microorganisms and sterilize the object or the surface. There are two ways to kill microorganisms in food using heat. One is by a process called ultra-high temperature, and the other is by pasteurization. In ultra-high temperature, the food is heated for a few seconds at temperatures that range from 135 degrees Celsius to 150 degrees Celsius. This process makes food last much longer without going bad. Although pasteurization does not kill all microorganisms in food, it is still a technique of sterilization. In the process of pasteurization, food is heated at 71.6 degrees Celsius for at least 15 seconds or 62.9 degrees Celsius for 30 minutes. The majority of the microorganisms are destroyed. However, since pasteurization does not entirely kill all the microorganisms, the food will eventually go bad. Pasteurization makes the food or liquid last longer and makes it safer to consume. Milk and beer are two examples of liquids that need to be pasteurized. Let us watch a short video clip about pasteurization.
It was Pasteur's work on behalf of the beer and wine industry that put him on the right track. Pasteur was trying to find out what was spoiling so much of the country's wine production. He discovered that the spoiled wine was contaminated by microorganisms, germs, and the germs were causing the wine to sour. But with a simple heat treatment, he showed that the germs could be killed off and the wine saved. The pasteurization process was born.
Welcome back, students. Let us move on with the lesson. The final technique of using heat to remove microorganisms is dry heat. Dry heat uses hot air that is free from moisture. The heat is absorbed by the outside surface and then passed inward until the object is eventually sterilized. This technique takes a long time, but removes all the cells. An example of how dry heat is produced is through a special oven used in biology labs that sterilize equipment. In our first activity, we explained that cooking your food is a way to eliminate the microorganisms that live in and on it. Can you list the names of the 2 techniques we learned about that are used to kill microorganisms in food? Please take a moment to write your answers on a piece of notebook paper.
Students, let's get ready. Begin. Time's up. Let's get back to our lesson.
Welcome back, students. The 2 techniques we discussed that kill microorganisms in food are ultra-high temperature and pasteurization. If you got these answers correct, well done. We have learned about methods of destroying microorganisms, but often scientists want to grow microorganisms so they can study how they look and how they function. When scientists grow microorganisms, they must use aseptic techniques. This means a procedure that is done under sterile conditions. An example of aseptic technique would be flame sterilization. Scientists grow microorganisms in an incubated environment, which is an environment that has been designed for the microorganism to flourish. A suitable temperature and the right nutrients are provided for the microorganisms so they have everything they need to reproduce. The final term we will learn about today is inoculation. This is where a microorganism is introduced into a culture medium. A culture medium is a liquid or gel that contains nutrients that provide the bacteria with the material needed to grow and reproduce. In our lesson today, we learned about how the method of heat is used in sterilization. We learned about 5 different heat techniques. Those techniques are boiling, autoclaving, ultra-high temperatures, pasteurization, and dry heat. We also learned about aseptic technique, inoculation, and incubation. Until next class, thank you, students. Thank you, teacher.